Okay, so good morning. We have Korzan with us today. It's a pleasure to receive Wayne here. We know each other for some years now, and we have always been working in closely related areas with very similar interests until recently Wayne decided to enter a new enterprise and join a lab working on orphan receptors in olfaction, and so trying to discover new ligands to these receptors in mice or rats? Mice. So there's one mice person here, and ever since before that, Wayne was interested in the social effects or the effects of the social influence and social interactions and social status on the organism and basically on the emergence of social phenotypes based on the experiences and the context in which the individuals are placed. And so Wayne got his PhD from South Dakota, working in Cliff Summers' lab with lizards and fish.
Anolis carolensis, yes.
Yeah, but mainly with lizards.
Mainly with the anolis.
And then he moved to Stanford to work with Russ Farnold as a postdoc, and then more recently to Harvard to embark on the mice stuff. Mm-hmm. And I won't keep going on. I just wanted to thank you and give you a short introduction.
All right, thank you, Rui. Thank you for those kind words. Yeah, I'm still interested in social hierarchies and mechanisms in the brain that are influenced by the behavior and back and forth the interplay between those two. But Rui's right. I'm taking a detour right now to work on try to find ligands for orphan receptors. But I'm not going to talk about that today. I'm going to talk about some of my work with Russ Farnold at Stanford University. And this talk has a very long title, but I think it's very descriptive, so I went with it anyway. And today I'm going to be talking about mRNA levels of some glucocorticoid and androgen receptor subtypes in this fish species and how they change with social status in the preoptic area of this animal. And some of the changes suggested, or to us, suggest that they might be neuroprotective. So before I get into the experiments, I just want to outline quickly what I'm going to talk about today. And of course, first I'm going to introduce the animal model, the system, so you guys get an idea about that, about the species, instead of Tony. Then I'm going to talk about this first experiment that's the major bulk of this talk, looking at stable hierarchy, where I paired males together. Now these animals normally live in large groups, but just with two males. And I'm going to show you some behavioral and hormonal results from that, and as well as quite a bit of gene expression data. And then the third thing, or the second experiment I'm going to talk about is social ascent. And these are also paired interactions and have some behavioral and hormonal data from this as well. And then I'm going to just make some all-encompassing summary statements about working with this model and where it's going to go for the Farnold lab in the future. So the model system, is an African cichlid fish, which lives in leek-like social systems. I'll explain that a little bit more in a moment. In the lake, in Lake Tanganyika in Africa, and the fish that we work on, the parent fish, the original founders of the colony, were taken off the coast, or off the shore, I should say, of Burundi, and in the shore pools. So the reason why this fish has gained a lot of fame at Stanford for Russ Farnold is that the males have a very interesting phenotype where they exist in either one or two conditions, either territorial or non-territorial, or dominant or subordinate. And we have a nice photo of a territorial male here and then a non-territorial male here. So they visually look quite strikingly different. And the cool thing about this system, the really interesting part of it, is that the males move back and forth between these two phenotypes based on social interactions. So if you win the fight, you either remain or become territorial. If you lose it, you become subordinate. So this system, and Farnold lab's been working on almost 30 years now. So we know quite a bit about the system itself. And the animals, actually, the males come in one of two different colors, either yellow or blue, and then also for the non-territorial animals. Now, the territorial males make up about 30% of the population. They defend these territories where they spawn with females and have access to food. Now, some of the physiology, they have very large GnRH neurons and thus make a lot more GnRH peptide, and then have very large testes. And just the opposite for the non-territorial males. Their GnRH neurons are actually 8-fold smaller on average, and then they make a lot less GnRH, and they have regressed testes. In fact, if they're non-territorial for long enough, they don't even make functional sperm, even though they're adults. And these animals school in the open water with the females, and hence the leak-like social system, and they make up a majority of the population of males. So, because the territorial males have large functional testes, they're making higher amounts of testosterone and 11 ketotestosterone androgens, and their social status of being aggressive and not being beat up, they have low cortisol levels at baseline. Now, just the opposite for the non-territorial males. They have low androgen levels and they have very high cortisol, presumably because they're being beat up on all the time and chased. But they grow faster, and because they grow faster than the territorial males, there's this natural cycle of being territorial for about 3 months and then non-territorial for 3 months, back and forth. At least that's how it is in the aquarium. In the wild, it probably could be faster or slower. I don't think anybody's actually looked at that yet. So before I get into some data, I'd just like to show you two quick videos. And this first one is kind of a treat for us, because rarely do we see or catch on film a non-territorial male coming down and challenging a territorial male, just because we're not filming them 24 hours a day. So I just so happened to catch part of one of these fights, and I'd like to show it to you. And the yellow male has come down from hanging out with this other non-territorial and these females up here. Now he's going back up there. But we catch the video when they're just doing a side display, and the yellow male goes and bites the blue male. His territory is here, and he wasn't quite aggressive enough to make the blue guy give up, so he goes back up with the females. So they have a very dynamic behavioral response to each other. And while this is happening, you can see that this blue male and another yellow male are actually fighting at the same time. So there's lots happening in each one of these tanks. And the tanks will have anywhere between, depending on how big the tank is, 3 to 5 to 6 territorial males, and then lots of females and non-territorial males both are schooling up at the top. I didn't say this when I started, but if you have any questions as I'm going along, feel free to stop me. So the next video I want to show you is what the animals do most of the time. So we have territorial males. What the territorial males do most of the time, they do this border display where the males come together in an imaginary border, the border of their two territories, and they face off and do this back and forth motion and turn sideways and show the other animal how big they are. And while they're doing this, almost always the schooling fish will come down and try to find food, and while the cat's away, the mice will play kind of situation. So during this video too, you see a nice example of a non-territorial male here, and so there's a striking difference between the coloration, and they can turn that on quite quickly or turn it off quite quickly. The other thing that the territorial males have that's very obvious is this eye bar, and they can turn that on in milliseconds, and it's a signal of aggression and aggressive intent. So what I wanted to do is, there's been many different paradigms used in the lab over the years, and I wanted to try a different paradigm because all the variable ones that we've had, I thought that it would be really important to get very stable non-territorial and territorial males. And so what I decided to do was take a tank and one of our large tanks, divide it in the middle with a clear perforated divider, and set up essentially 2 experiments in one tank, and just put 2 size-matched territorial males together. And when you do this, almost immediately, within the first few minutes, up to an hour, one becomes non-territorial, and then that's it. He will stay that way for the duration of the experiment. And we left him like this for 1 month, and behavior and status was recorded for the first 3 days, and then the last 3 days. But the status was checked of the animals every day, but we recorded the behavior. on the first and the last 3 days. And immediately following the last recording, the animals were sacrificed and their plasma and brains were collected. Now, in our lab and other labs, just to look at animals' dominant behavior or a dominance index, we use this measure, this dominance index, that is basically all the dominant behaviors, aggressive behaviors and courting behaviors, minus all the subordinate behaviors, retreating, hiding, fleeing, things like that. And just to verify that at the end of the 1 month, we still had a nice separation of the hierarchy. The dominant animals or terrestrial animals are clearly more aggressive. And this is a very low level compared to what you see when you first put them together. The numbers would be 10 times higher or something like that in the when they first start interacting. But we have a nice separation. The animals are clearly territorial and non-territorial. So I've told you about the androgens that we've measured in the past. You need to check those. If some of you may not be that familiar with androgens, but I'm assuming that all of you are because you're working with Rui. So testosterone and 11-ketotestosterone are really important for regression, as we know. And they're also important for secondary sexual characteristics in this animal. And a good example of that is these egg spots that you see that are nice and big and pronounced on the territorial male and very small on the non-territorial male. And these are directly correlated to how much androgens these animals have. Now they use these for spawning, these egg spots. It's part of the spawning behavior where the female comes into the territory, she lays her eggs down, picks them up in her mouth, because they're mouth brooders. And when she's doing that, he lays on his side and extends that fin where her eggs were, and she tries to pick the eggs off of his fin, and then he releases the sperm. So these two androgens are really important for these animals. And also, the other hormone that I'm going to talk about is cortisol, and because social interactions are stressful for the one that loses. So cortisol is something I'm going to talk about, and of course it has other purposes besides the stress response that are also important for growth and general metabolism of the animal. Just a brief summary on testosterone and its relevance to this talk. It's the primary androgen in invertebrates. It has a multitude of behavioral and physiological effects, some of them beneficial and some of them detrimental. And on this figure to the right here, essentially have a lot of the detrimental effects on the left and beneficial ones on the right, such as increased injury, suppressed immune system as being sort of bad, spermatogenesis and muscle hypertrophy good. And because it's this double-edged sword of testosterone being good and bad, most species, vertebrate species, only increase the levels during reproductive important times. And this decreases the amount of detrimental effects, if you will. So for this talk, mostly we're going to center around the testosterone's and 11-ketotestosterone's effects on sexual behavior and more specifically aggression and territorial behavior. So there is some differences between testosterone and 11-ketotestosterone down to the receptor level, to the point that androgen receptor subtypes in many of the systems actually have a higher affinity for 11-ketotestosterone than testosterone. And Rui is a great person to make the statement. It's okay. About that there's a strong link between 11-ketotestosterone and reproductive processes, because many papers showing that. And we also see it in this system. So what do they look like in my very stable hierarchy that I've created? Well, as we hoped, the testosterone in the territorial males is much higher than the non-territorial males, and as well as the 11-ketotestosterone. Now we've seen this before in other experiments from the Fernald lab. The nice thing about this particular experiment, actually the effect is even larger than we see on average in the lab. So the behavior looks good, the androgens look good. What about the cortisol? So the socially subordinate individuals in many species usually have elevated levels of cortisol. And this response is adaptive because it usually allows them to get away and take care of themselves. But if you have a chronic activation, if the animals can't escape, this can be very detrimental to the animal. Excuse me. And this chronic exposure because of the way these animals choose to live in these little societies, we think is one of the main a key factor in how they're repressing the reproductive system of the non-territorial males. So, and that's been thought of in the Fernald lab for 20 years. So that's no new surprise. So the cortisol looks great. Actually, it looks a little bit better than a lot of the other studies we've done, but the pattern is beautiful with the non-territorial males having significantly elevated levels. So now we have behavior and the hormones look great for this paradigm. So let's look at some gene differences if there is. Now, one of the a hot topic in one of our lab meetings was, if everything's so stable, would you expect to see any differences or any changes in gene expression between these two statuses? And that's a good point. And so went into this with a little bit of being nervous that maybe we wouldn't see anything wherever we looked, because it's so stable and nothing's really changing for these animals. So with that being said, more, for our system, where and what do you want to look at, and how are you going to do this in these stable animals? Well, the where for these guys is very easy. The GnRH neurons are located in the preoptic area, so that's where we'll go first. How we're going to microdissect out the POA, which is something I'll get into a little bit later about the microdissection, but something we're going to be doing here in Ruth's lab in the next couple of days. And what? So the hormones that I'm interested in, and some of the key ones are androgens and the glucocorticoids, so we'll look at their receptors and see what the mRNA levels are of those. So the preoptic area. In this fish, this is a side view of the brain, and this C line here corresponds to this panel below, and this is the preoptic area. These black, dark, very large neurons are GnRH1 neurons in a territorial male. And this is a key nuclei for this model, as I've already told you, because of the GnRH production in this area. And we also know from in situ data that there's glucocorticoid and androgen receptors that are located, co-localized with the GnRH1 as well as in the surrounding area. So, how do you get this one area away from the rest of the brain with some kind of savvy? The problem with trying to work on one nuclei and trying to work on just the GnRH neurons, which we would like to do, is you could try to microdissect out the small area, or you could do something like laser dissection, but this is a very low-tech way of going about this. So you punch out the area using a micro punch, and but before you do that, actually, you remove the brain. You want to freeze it and then slice it frozen, mount it on a slide, and then we use 300 micron sections, but you can change that a little bit, and then punch out the brain and put it into your desired buffer, whether it be Trisol to break them down, or another kit from Quieten or something like that to prepare the RNA, and then prep it for analysis for quantitative PCR. That's what I did. So the first thing I wanted to check was to just verify that I did indeed get the GnRH neurons. And so when I ran the quantitative PCR for GnRH1, the mRNA levels indeed are twofold higher in territorial males than non-territorial males. And when I sampled areas around the POA, I didn't see any mRNA levels for GnRH1, so got the right area. So this is more of just a control saying, indeed, got the right area, and we didn't really miss any of it. Well, what do the androgen receptors look like? I showed you what the hormone data looked like for this situation. And what we saw was a significant increase in AR alpha mRNA levels in territorial males compared to non-territorial males, but we didn't see a difference in AR beta. Now, the AR alpha is more sensitive, has a higher activation or higher affinity, I can't remember which one, to the androgens than the AR beta. So this suggests to me that this data just says that AR alpha is probably the one in the preoptic area that's facilitating the positive feedback of keeping the GnRH being produced. More GnRH is released, more androgens in turn come from that. So that's what that's what we think about that. But now, let's move on to the glucocorticoid receptor story. And it's a little bit more confusing, a little bit more, not confusing, but a little more complicated. So in Teleos, this nice diagram here by Stolt et al. In 2006, that I really like using, has the GR, the glucocorticoid receptors for Teleos divided into two main groups, a GR1 and a GR2. And then down here they have the mammalian ones, and also frogs, chickens, and things. Excuse me. In the humans, there's actually three types that have been found. Beta is an interesting story. I can talk with people about the specifics of the human part when they're interested, but the alpha one is the primary functional receptor subtype, and not very much is known about the other two. But in Teleos, we have the Ratuni mouth brooder is actually the fish we work on, is Tilapia ratoni. But we discovered these three, the GR21 and 2B, which is actually GR1A and 1B. And I changed the numbering later on. But we were the first lab to actually find that there's three subtypes. So we actually mislabeled them. So everyone else is calling these one, and we called it two. But since they were the first lab, now when we go back and publish later, we have to fix that. But then, excuse me, I'll talk about the differences in what we saw in the second here. But first, let's just show you the in situ data for looking at the localization of these receptor subtypes. Now, I didn't mention that there's MR, also mineral corticoid receptor, found in the species, but this actually doesn't look that great. But anyway, I'll tell you what you see on the computer. The silver graining, or the white spots, are silver stain signal, and the darker splotches are GnRH1 positive cells. So the MR and the GR2 are kind of interspersed. The glucocorticoid receptor number 2 and MR are interspersed throughout the region of the preoptic area, not really co-localized on the GnRH neurons. However, GR1 is very co-localized with the GnRH neurons. So then this is just a sense.
This is hybridization or in situ hybridization?
Yep, in situ hybridization. Anna Greenwood did this in 2004. And to get a little bit more at the heart of other things Anna looked at with these glucocorticoid receptors, she analyzed what the difference was between the GR1 alpha and beta subtypes. And the only difference between the two is that the GR1 beta has a 27 base pair insert in between the zinc fingers of the DNA binding domain. So the cortisol binding domain is intact. It's exactly like the GR1 alpha. The only difference is the binding domain to the DNA for the second subtype. And she went on to do a cell culture analysis of activation of this receptor, putting it into a vector and transfecting it into the cell line, and then testing to see the activation with a range of hormones. But we're going to mostly just concentrate on cortisol here. And what she saw was that GR2 and GR1A have very nice activation of, in the presence of cortisol, but 1 beta has a much reduced activation level compared to its splice variant isoform, and then the other main glucocorticoid receptor. And we noticed that also, just as a side note, MR bound aldosterone or cortisol with the same kind of activity. And to my knowledge, I don't think anybody's found aldosterone in fish yet. I don't think it actually is there. Anyhow, so this MR receptor might be important for Teleos for other reasons. It may still be a functional receptor, just with a very low affinity. So one thing that you think about just with one concentration of glucocorticoid, maybe the difference is just in the affinity, or maybe it's an affinity problem. But when you look over a course of different concentrations, you see that the pattern still remains the same. GR2 being the highest and the best activator with cortisol present, and 1A very similar, and 1B highly reduced. So what did we see in this stable situation? Well, a little bit to my surprise, the MR MR and the GR2 receptor subtypes were significantly elevated in territorial males compared to non-territorial males. And as I said, the MR receptor, we don't really know a lot about this particular part of the story, but there's actually an undergrad looking into this. And so, because it may have playing a role in aldosterone. Now the GR2 having a higher amount in territorial males compared to non-territorial males a little bit surprised me, because this is the high activation, high affinity receptor subtype, but maybe that's a good thing for these animals because they have such low amounts of cortisol available to them. So it would be beneficial for them to have a very good receptor to take advantage of flow levels. And kind of cruising through here. Okay, so what does the 1A and 1B look like? So for glucocorticoid receptor 1B, we didn't actually see a significant difference between territorial and non-territorial males, but there's definitely a trend towards that. And when we looked at the 1A plus B, we saw actually an increase in territorial males. And if you're wondering why we did this the way we did, is because you can't separate 1A away from 1B with quantitative PCR analysis, because it's essentially the same gene except for this 27 base pair insert. So you can separate out 1B though from 1A, and then you have to figure out a way to show this data that has some kind of meaning actually. Because these look in stark contrast to each other, the way we look at this data is to look at a ratio, which is in this graph. And so we're interested in what are the levels of 1B compared to 1A plus B. And what we see is that when we look at the ratio this way, we see a significant difference in the non-territorial males. And you may ask yourself, well, why would you go through all this effort? One of the really interesting things about the animal, like I said before, is the GnRH neuron size is so different. So if you're working with a very large neuron, it's going to have the potential to have a lot more space inside of it for receptors. And since the animals really only have about 200 neurons, the more space you have, the more receptors you could have in there. And that's why you see more really of 1A plus B in the territorial males, but it's just the opposite for the 1B subtype. So this is pretty exciting actually for us, because to be expressing a large amount of a receptor that isn't very good, it seems counterproductive at first when you think about it. But if you're an animal that has a lot of glucocorticoids in your system, maybe it's actually a good thing. And I'll try to use this small schematic that I put together to try to tell my story a little bit better here. So if you imagine that this is a territorial male's GnRH neuron, it's very large, it has MR, GR1A, and 1B, and GR2 receptors in it. It has a low amount of cortisol available for its present, and it makes a lot of GnRH1. And so the cortisol that's there would be beneficial to have receptor subtypes, especially GR1A and B, that are really good at binding and signaling glucocorticoids. However, for non-territorial males with their very small neurons, they have fewer of these good receptors and a lot more of this very bad receptor. So, but they have a lot of glucocorticoids available to them. And when you think about the glucocorticoids entering the cell, if you know the literature pretty well with glucocorticoids, high amounts of glucocorticoids kill cells. And for these animals, you don't want to kill the cells, because you make this transition back and forth between territorial and non-territorial many times during your life. So if you don't have a way of protecting your neurons from high levels of glucocorticoids, there's a chance that some of them will be damaged or killed. So that's one of the hypotheses about why this is happening. The other one is a little more mundane, but it might be just this receptor subtype actually activates a whole different pathway, and actually is the reason why you have decreased cell size and decreased GnRH1 being made. But when we look at lots of other areas, important limbic areas, we see this pattern of 1B being increased in very important places like the hippocampus and parts of the amygdala. So to us, it looks more like neuroprotection, a neuroprotective mechanism. So you imagine that cortisol comes into the neuron, and this receptor subtype binds it up with the same affinity as GR1 alpha, but then doesn't translate. It doesn't do anything with it, just holds it there. So that's the way we're looking at that, and it's pretty exciting. It's a pretty exciting result, actually. But let me switch gears now. So now I've talked about this very stable situation. I also did an experiment where I wanted to investigate, well, how are these differences now that I've told you about, how are they happening, when are they happening? So Hans Hoffman had a very nice paper, and I really like this figure from it, where he looked at animals that were established non-territorial males and established territorial males, and then he tracked the ascension through the mean dominance. This is basically on this side 0 is dominant, on this side is subordinate. And when he gave them the opportunity to ascend, by the first day, their GnRH neuron size had already been significantly increased. By 1 week, it's quite even more, and by 2 weeks, it overshoots where it will, on average, end up. And what you see is that during his study here, the change is slow, but actually pretty fast. But going the other way, the behavior changes very quickly, and the neuron size decreases at a slower rate. So it's a little bit faster going up than coming back down. But I think this is a great figure to lead into the thing that I did. I wanted to use the same paradigm that I had before and look at ascent to see what the animals, at least for the receptor subtypes and other things like that, what are they doing? So in another study that was going down the same lines by Sabrina Burmeister, she was really interested in early genes, EGR1. And what she did is she took subordinate males and dominant males, and then recorded the behavior, just all the behaviors that the animals produced for the subordinate animals before they were allowed to ascend, and then the day that they ascended, and then animals that didn't get to ascend, and then just stable dominant animals. And what she found was pretty interesting. The variability, this is really hard to see actually, the latency to when they started this process of ascending, at least behaviorally, was very variable between, there's one here that was at 2.5 minutes and one at 14. So very different between the animals. But you also see quite a bit of variability in established dominant animals, where some of them took 3 minutes and some of them took 6 to start behaving for the day. And so it suggested to us and to myself that every day is a new day, essentially. So on every day, the animals have to reassert themselves in the morning. And she was, like I said, she was interested in EGR1. So she has this very nice figure where she looked at the number of GnRH positive cells across her sections, and she had 10 sections. I think there were 14 microns each. And then on this other axis, she has the silver green stain or signal for EGR1. And there was a direct correlation. The more GnRH neurons there are, the more staining she saw. And she saw a very nice co-localization between GnRH1, which are these brown guys, and you can't see the silver staining at all, but it's there. And so this was a nice starting off point, and she did this at 20 minutes. So she let them ascend for 20 minutes and then took these tissues. So I want to use a very similar paradigm as I did before, so I can hopefully compare the two experiments.
At 20 minutes, she had the EGR1 signal there already?
Yep. She had a significant increase in EGR1 in 20 minutes. So so I had a very similar situation as before, only this time I used a male 20% smaller than another territorial male, so I can make sure which one would go down. Then NT, and what happens is as soon as you put them together, of course, the smaller male turns into a non-territorial, And you leave them like this for, I left them like this for 3 weeks, or I'm sorry, for 30 days. Sabrina only did hers for 2 weeks, but otherwise they're very similar. And then recorded the behavior as before. And then 1 hour before lights on, I went in the dark with the infrared scope and removed the territorial male. And as soon as you do that, when the lights come on, at some given point, the non-territorial male becomes territorial. He starts fighting with this control guy over here and starts courting the females. And as soon as the 45-minute time span was up, I killed the animals and took the brains and blood. Now, the first behavior that you see out of these animals when they ascend like this is this eye bar. And they turn this on the first thing, even before they turn on their other colors really brightly. And they have neural control over this, so they can turn it on and off in milliseconds. It's direct innervation from one of their facial nerves. So, and it's a really cool story actually. If you cut the nerve on one side, they'll only show the other side to other animals, even though they can't turn it on. So they're very conscious about their reproductive success. But so I just wanted to quickly compare the territorial males. This is territorial males. Every day that they wake up, they're territorial. These are very stable males, and it takes them around 380 seconds to turn this signal on, and then start chasing the females and beating up on the non-territorial males. Now, males that are ascending take about twice as long to do this, but it's just to me it's just remarkable that they only takes them twice as long actually to do this because they've been suppressed for 30 days. They haven't had their eye bar on for 30 days. So that was a cool result from the ascension. Now, the other really cool thing about the animals that ascended was that if you looked at the last 3 days and averaged the amount of aggression they received on those 3 days and looked at how long it took them to turn this eye bar on, there's a very nice correlation between how much aggression they received, how beat up were they over the course of the 3 months, and how fast they came out of it. So that was a nice result. The other thing, even though I due to experimental issues, lost quite a few of these animals, there are also the animals that I was able to get blood samples from. The 11 ketotestosterone for the ascending males had a nice correlation, direct correlation to how long it took them to ascend also. So if the males were able to hold on to a little bit more of their testes and make a little bit more androgen, they were much faster at ascension when the opportunity arose. And these obviously were also the same animals that got beat up less. So they weren't driven down as far. So we see nice patterning of how much aggression, how much of that will affect, to what degree, the physiology. And the next slide. So we also, of course, also measured cortisol and compared it between the non-territorial males that did not get to ascend and the ascending males. And a little bit surprising at first until you think about it, the ascending males had significantly elevated levels of cortisol compared to the non-ascending males. Now, if you saw the animals, you might not be surprised because when they start to ascend, they are just absolutely crazy. They're chasing the females, trying to court with ones that aren't interested in them, fighting with the guy across the barrier, and just flying all over the place trying to establish themselves. So they increase their locomotion by 200-fold. So having more cortisol shouldn't be that surprising because they're really, really burning up resources. So 45 minutes of this, their cortisol levels are really high compared to these animals who are basically hiding from their territorial male. So the other thing that in the preoptic area, so I also removed the preoptic area and then ran it for the different genes. And one of the first ones I wanted to look at was EGR1 because at 20 minutes we know it's elevated, and at 45 minutes it was also elevated compared for the ascending males compared to the non-ascending males. The only trouble I have with this data, even though I didn't add the graph, is that if this was a territorial male and this was a non-territorial male, the pattern is exactly the same. So in the stable situation in the preoptic area, the pattern looks like this. In the stable situation, it looks like this. At 20 minutes, it looks like this. And at 45 minutes, it looks like this. So I'm not quite certain if I could have accidentally hit just the right times, or that just EGR1 is on in these animals if they're either ascending or territorial, just more. But it kind of makes sense too a little bit, because the territorial males have these huge neurons and making lots of GnRH and making lots of stuff. They're probably just, the tone of them is just active. The non-territorial males are trying to shut everything down. So all the other genes that I showed you before, and I measured 20 different ones in each sample, I didn't find any differences in the other 22 genes that I tested 45 minutes after lights on. So back to this figure, we know that by 1 day, there's a significant increase in the soma size, but between both Sabrina and I, 20 minutes is not long enough to see any difference in at least the cell size, and EGR1 seems to be on at that time. But the other factors that I hoped to investigate may be just a little longer. So it might be just a temporal issue. If I looked maybe at 2 hours or 3 hours, but trying to figure out what time point are these changes starting to happen. And maybe it's not even in the first day that I would see the same things that I've seen in the stable hierarchy. Maybe that happens at 1 week, maybe that happens at 2 weeks. So there's some experiments going on right now in the lab that are trying to figure out, well, what order of things are changing, and what order are things changing, and who's coming first, and what is the temporal pattern, who's starting, what's the next thing. Because now with the results that I have for the stable situation, we have at least a lot of key molecules and key things to look at to try to figure out when are these things changing, and who's coming first, and these kinds of things, and which part of it is either activating the growth of the neurons, or which ones are restricting it. So just to make some overarching summaries, this species actually, even though it's genetically a little bit cumbersome to work with because the genome isn't fully sequenced, the behavior and the other parts of it make it a very interesting and dynamic species to work on. And it allows a very neat way of looking at social interactions and how they affect physiology and neural mechanisms. Now, the characterization I've been able to do with a lot of the things at very stable situations has really helped us out and given us some places to look at some things to try to figure out as far as how the behavior and the molecular stuff and the interplay between those, what's going on during the change and the physiology. And in the future, which is right now, I've left the lab now for a year, and the people in the lab are coming up with some good data trying to figure this out. So they're making quite a bit of progress looking into what kinds of things are changing first, and those kinds of questions. And of course, you can't have a talk without acknowledging and thanking people that helped you. So at Stanford University, Dr. Fernald and Dr. Zhao and Dr. Chen helped me learn how to work with the animal and different aspects of molecular techniques, because when I walked in I had none. And then also I'd like to just give a quick thanks to Stephen Lieberless, my current PI, for letting me have this week to come and be with you guys and tell you a little bit about some cichlid stories. So with that, I'd be more than willing to take any questions that you may have. Was that good, huh? No questions?
Absolutely. No questions? Can we go back to the cortisol level just to...
For the ascending ones or the
stable ones?
The stable.
And these levels relate to the levels when you have...
When they ascend?
Yeah.
You have a keen eye. Yeah, they're dramatically different, actually.
Yeah.
And the main reason, I think the main reason, actually, is the time of day that I collected them.
Okay.
So the ascension is happening 1 hour after lights on, when the animals are the most active. And their cortisols are, cycle natural rhythms just like every other creature. So they're naturally a lot higher in the morning. These animals were collected at 3 in the afternoon. So their levels...
Because the other levels are much higher.
Oh, much higher, much higher. And I've actually done a study there with the animals looking at territorial and non-territorial males or have everybody be a territorial male and have them individually housed, and then take a blood sample at 3 o'clock and then pair them up, and so one becomes a non-territorial male, and then do it 3 o'clock, 9 o'clock, 9 o'clock, and 3 a.m. So 24-hour in the same males, which is really difficult because sometimes they die. But I did get enough data from that to see that the morning is very. If you do it before right when the lights turn on, they're still a little bit lower. But if an hour after lights on, they're really going up fast because their animals are the most active in the morning, and they are that way in the wild too. In the wild, well, this is what I've been told. I've never seen them in the wild. Never been to Africa, but in the mornings it's pretty calm, and the animals do a lot of their stuff for the whole day in the morning, and then by the afternoon the winds pick up and turn and turbulate up the lakes, and so by the afternoons they can't really see, and these are very visual oriented animals. So yeah, I was going to put them together, but they're so different that and I really can't. I wanted to compare these two experiments in a paper, but I just can't. I mean, I could talk about them as two individual experiments, but the caveat of the cortisol being so different and the time of day being so different, it's not very, it's not adequate. It's not even a favorable comparison.
And do you have any idea of how these relate to their maximum levels?
Oh.
So if you do that's very similar to issues that we are going through here in the lab. So
yeah.
have you considered that doing a ACTH challenge and see how much cortisol you can get out of these guys and how these levels relate to the maximum physiological level?
This has been done. Russ somebody has done this in the lab, and what they did is they did a restraint stress for the top physiological one, and then they did, I think they did do an ACTH challenge too, but I can't remember what the numbers were. Okay. And I know that as far as like physiological limits, the status is pretty close to like even restraint stress. It's not an order of magnitude or anything different. It's in the same area. Of course, restraint stress is higher. But
what do you call restraint stress?
Net. Yeah.
And how many minutes?
Oh, these fish are robust, so I think they did
10 minutes.
I think they did 10. I can't remember, but it was Tricia Tricia Clement did this with a net stress. I can't remember.
Restrained in the water. They were not.
I think they were out of the water
for 10 minutes.
These animals will fall out of the tank and or jump out of the tank. They don't fall. They jump out all the time. And so we have covers over them, but we have holes so we can feed them and have air stones going down these holes, and they'll still jump through the holes. And they'll fall on the floor and they'll dry out. Like they'll be hard.
Yeah.
You throw them in the water and they come back. I mean, they won't be breathing for minutes. They'll be completely dried out on one side, hard.
Mm-hmm.
You throw them back in the water and they'll sometimes come out of it if the other animals don't eat them first. So they're really, really tough. I mean, they can really survive a long time without water. I mean, for a fish, a long time without breathing.
Because then the next question is that the neuroprotection hypothesis assumes that that these levels are already having, negative impact in terms of either neurogenesis or survival or whatever.
We see that. I mean, they're high enough where this chronically, I mean, they're not the peak, but they're high enough to the point where sustained over, the only reprieve they get is at night. And, but they're high enough that their scales are falling off, they're shriveling up. They'll change, actually. This is really interesting too. The body size changes. Non-terrestrial males in that status for 1 month in my paradigm actually shrink 8% in length, and they lose like 30% of their weight. I've published this, but I can't remember the exact numbers, but they lose a ton of weight and they shrink in length. And there's only a couple species that are known to do this, and one of them's the Galapagos iguanas.
Mm-hmm.
They can survive. This is a science paper, I think, where they actually, some of the animals shrink in length to adapt, to have a smaller body size, to adapt when times there's not very much food. And these animals will do it too. I checked the measurements twice, 3 times because we couldn't believe it. But the terrestrial males gain weight, and the non-terrestrial males lose weight and shrink. So, and they look like hell. I mean, they look really bad at the end of a month. And but you have some in the aquarium, and then the next question is, well, is that not physiologically relevant? In the way they're in the wild or in the way that we house them. But some of the animals in the way we house them do look like that, but they don't look like that for very long because all the other territorial males just beat it till it dies. And these animals kill. Well, cichlids. I mean, they kill each other like crazy, so you really have to watch how, you have in a tank, you have to have a certain density of this, they kill each other. If you don't have enough, one of them kills everybody. So they're hard to handle. But, the whole point of doing the stable thing for that long was I wanted to push to the edge of the physiological relevant edge. Whatever they could handle, that's what I wanted to take it to the edges. So we might make the rest of the data a little cleaner, or at least make it easier to make some predictions about it.
And if you pursue this idea of neuroprotection by looking into the mechanisms,
Russ and I just had a meeting last month that we talked a little bit about this, and he has a postdoc that's very interested in I'm actually taking these receptor subtypes, putting them in a cell line, a neuronal cell line, and testing to see if, at what levels, like, do the cells die, cortisol, what kinds of, how neuroprotective is it? Is it neuroprotective? It's one way to ask that question. But I've moved on, so I don't have any access to do these kinds of things anymore right now. I think something in the back of the question are sort of where my hand almost. Maybe not.
No. Well, Gwen will be with us for a week. So there will be plenty of opportunity to send us these questions and discuss issues. I think there's a number of parallel stories going on here that people could talk to.
Yeah, I would be more than happy to. It's a pleasure to be here.
I think that's it. Thank you again.
Yeah, thank you for all coming.
We have a very short kind of planned meeting.
Oh, okay.
But during this week there were these grant applications going on, so we had a chance.
So
just
do what I do.
Do what? And instead we talked to people for like 10 minutes.